So, all right. So now let's talk about how we can deliver the drugs. So lipids are very good a system to deliver the drugs. Okay, so they can be useful as a bioactive molecules for drug delivery. Okay, so either we can use it through a nano capsule and a nano capsule. It's called a liquid core containing drug, or a nanoparticle, a solid lipid structure. Okay, so lipids can be used because they are non-toxic, and they are non-toxic. They are very stable means of administering the drugs, and especially you can control the rate of drug release. Okay, so here is one example to show how we can use a nanocapsule or nanoparticle. Okay, nanocapsule. In this nanocapsule, this is a liquid core containing the drug. So these are drug molecules, and this is a shell. And usually these are loaded with the oil. Okay, so the drugs are loaded with an oil, so then they can be useful as a nanocapsule. Okay, and whereas in the case of nanoparticle, they are useful as these are solid matrix. Okay, and these drugs are dispensed or dissolved in the matrix, and then the drugs are absorbed to the nanoparticle, and then it can be released. Okay, or it can be delivered. Okay, so these are two different ways you can actually deliver the lipids: nanocapsule and nanoparticle. These are lipid-based drug deliveries. Okay, let's talk about membrane lipids. Okay, so where they are and how they behave. So usually 5% of the genes synthesize all these lipids. Okay, various glycerolipids, sphingolipids, and various types of lipids. Okay, and usually they are dependent on the complex lipid repertoire. Okay, so and what we do understand the specific functions of numerous lipids, but for many of them, their functions is still very, very unclear. Okay, and we can use mass spectrometry, based approach to study the proteomics, the protein composition of those lipids, where they bind to and what they interact with and stuff like that. We can also use the genomics-based approach also to look like transcriptomics, to study the transcript level changes. Okay, so now there are also new disease-related genes out there that are involved in lipid-related proteins, such as enzymes like lysosomal hydrolases or ATP binding ABC transporters, ATP binding cassette ABC transporters, or the lipid binding proteins like the start domain protein. So these are various proteins, and those are lipid-based proteins. They have some roles in the diseases. Okay, because these are disease-associated genes. Okay, so let's talk about a little bit more about where is the main site of lipid synthesis. Okay, this is very important. Okay, as you all know, the endoplasmic reticulum is a major component. Okay, so the main lipid biosynthetic organelle that's where it's being synthesized is the endoplasmic reticulum. So the majority of the bulk of the structural phospholipids and the cholesterols, or the ergosterol in the yeast, they all are being synthesized in ER, or endoplasmic reticulum. Okay, and the significant levels of triacylglycerol and the cholesterol, the cholesterol esters, they all are having non-structural roles. Okay, but the bulk of the structural phospholipids and the cholesterols, all those things are being synthesized in the ER. Okay, the ER also produces what they call ceramide. Okay, which is a precursor for complex sphingolipids. Okay, and also what we call them as a GAL, the ceramides. Okay, these are so hold on a second. So where is this? Yeah, so the GAL ceramides is produced in the ER of the epithelial cells, and then usually they're important in stabilizing the myelins and the apical membranes. Okay, so the majority of the lipids, so the two important points: one, the structural phospholipids and cholesterols are all synthesized in ER, and the ER also produces the ceramides, okay, which is the precursor of the complex sphingolipids called the the ceramides. Okay, and see here is the 1 second. I just want to see one important thing. Just again a minute. I want to see. Just hang on a minute, okay? I think I. Yeah, so these are galactosyl ceramides. Okay, the gal ceramides are galactosyl ceramides. So, okay, so the main lipids are, the structural phospholipids and the cholesterol, so the ER, and the ER also produces ceramides, which is a precursor of the complex sphingolipids called the galactosyl ceramide. Okay, these are produced in the ER of the myelinating and epithelial cells. Okay, so now the main set of lipid synthesis, okay, the mitochondrial associated membranes, are a subfraction of the ER that co-fractions the mitochondria. So basically, the main set of lipid biosynthesis are called the MAMs. These are called the MAMs, okay? So MAMS, okay? Mitochondrial associated membranes. And these membranes are between, these are subfraction of the ER, but they are on the contact sites, okay? ER mitochondria and these MAMs are present there, okay? So mitochondrial associated membranes, okay? So these are called MAMs, okay? All right, so now they are particularly enriched in a specific lipid biosynthetic enzymes in both the mammals and the yeast, okay? Suggesting that the lipid synthesis may be subcompartmentalized in the ER. Okay, as you know, ER is the main site of cholesterol synthesis. The lipid is rapidly transported to other organelles. We'll talk about in a subsequent slide, how these are being done, which organelle goes where, which lipid goes to which organelle, okay? The ER harbors minor lipids that function as both pathway intermediates and legitimate pathway and products. For example, diacylglycerol or cytidine, diphosphodieselglycerol or phosphorylacetylamine, or lysophospholipids and dolicol. Okay, these are different phospholipids. Okay, so here are some examples to show in which organelle in which lipids are distributed. We know that distributions are not symmetrical, they are asymmetrical. So here's one example to show the lipids, how the lipids are moving from the endoplasmic reticulum and to the Golgi, to the plasma membrane, into the endosomes, okay? And usually the intrinsic lipid transporters dictate the phospholipid distribution across the bilayer. So now we will talk about each one of this organelle and how these lipid distributions are taking place, okay? So in the case of endoplasmic reticulum, if you look at it, these are a non-specific transbilayer equilibration of the phospholipids. Okay, so basically, there's a near symmetrical distribution between bilayer leaflets. Okay, for example, PC to PC, P to P, PS to PS. Okay, so these are cytoplasm, endoplasmic reticulum, and the side of cytosol. So the distributions are between cytosol and the ER, how they are actually going. So this is actually, if you look at this one, the arrows are going in both directions. You see there. That means like, there's a equal symmetrical lipid distributions are there, and there's equilibration is there of the phospholipids between two different compartments, ER to cytosol. Okay. Now, in the case of Golgi, it's a little bit different. Okay, these are not symmetrical. Okay, but these are asymmetrical in some fashion, because in the Golgi, if you look at it, the P4 ATPase translocate this phosphorylserine, which is called PS. So you can see the PS, okay? The PS and the PE, the phosphorylethylamine, is facing to the cytosol. So this is the cytosol, outside, this is cytosol. So now this is facing towards the cytosolic phase, okay? But the sphingomyelin is produced by the enzyme called sphingomyelin synthase so now if you look at this one, this is coming from phosphorylcholine. The phosphorylcholine, in both ways, it can go diacylglycerol or ceramide, but the sphingomyelins are actually formed from ceramides. Okay, you can see that the arrow is going only in one direction here. Okay, so Now the question is, neither phosphatidylcholine nor the sphingomyelin, okay, or in the ER, okay, but they are actually so non-sphingomyelin molecules that are resident in the luminal leaflet are transported to cytosolic phase because they are not transported in the cytosolic phase, okay, because that's why there's an X there here, you can see here, okay. But now there's also one more thing called but the diacylglycerol, yes, they are, okay. So if you look at this one, so here is what it says. So asymmetry in the Golgi is generated by the specific transport of phosphatidylserine and phosphatidylethanolamine and the lack of transport of sphingomyelin and the phosphatidylcholine. Okay, so so basically what it means is that the phosphatidylcholine is converted to the diacetyl. So the PC is the phosphatidylcholine. Okay, so this is the phosphatidylcholine, the PC. Okay. The phosphatidylcholine is converted to the diacylglycerol. You can see that the arrows are going in both directions because this freely equilibrates, okay, because that's why the arrows are going in both directions, okay. But now this diacylglycerol can serve as a substrate for the Golgi choline, okay. So there's a conversion of PC to diacylglycerol, and the diacylglycerol will form as a substrate, and that's why it goes back to the PC. Okay, so there's a both way. There's an equilibration is happening here. So, unlike the ER here, what they're trying to show here is that the phosphatidylserine and phosphatidylethanolamine, they are transported to the cytosolic phase, but whereas in the case of other phospholipids, okay, the galactosphingolipids, or the sphingomyelin or the phosphatidylcholine, they are not transported to the cytosolic phase. They are all present only in the luminal leaflet of the ER. Okay, so of the Golgi. Okay, but there's also, you can see conversion of the PC to diacylglycerol, but then diacylglycerol forms as a substrate, and we can convert them into PC. Okay, so this is how the general distributions are happening in the lipids, especially in the Golgi, okay. So again, keep in mind that this PE translocation from the Golgi to the cytosol is happening with an enzyme called P4-ATPases, okay, that can translocate the phosphatidylserine and phosphatidylethanolamine to the cytosolic phase. Okay. Now, in the case of plasma membrane, it's a different story. Okay, so here, what's happening? Okay, now you can see there the direction has changed. Now the direction is going inside. Okay, which means the plasma membrane is outside here. The cytosol is inside. So now the PE is pushing towards the cytosolic phase. Okay, like as we saw in the Golgi. Now you can see the PE is going in the cytosolic direction. Okay, but the P4-ATPases will support that. Okay. Now, however, the other phospholipids like phosphatidylcholine, sphingomyelin, and the galactosyl sphingolipids, they all are being disrupted because there's nothing, there's no translocation of the sphingolipids to the cytosol. Okay, this homeostatic distribution can be disrupted by the activation of the enzyme called scramblase and the inhibition of P4-ATPases. So once you have that, then this particular sphingolipids cannot get inside the cytosol. Okay, all right. So now in the case of endosomes, okay, so the phosphatidylcholine and sphingomyelins and the glycosphingolipids. Sorry, it's not it's glycosphingolipids or glycosphingolipids. Okay, so this glycosphingolipids, so they all were shown to be restricted to the luminal leaflet by a lack of specific transport mechanism. So here also the P4-ATPases are usually recycled to the endosomes. So basically what they're trying to show here is that in these endosomes, okay, these particular phospholipids, okay, are just present in the luminal leaflet, but they're not exposed to the cytosol at all. Okay. So you can see now, there are various lipid distributions. We talked about 4 different organelles, and you can see always, some phospholipids are exchanging and translocating to the cytosol. Some are not. Some are present only in the specific organelles here. Okay. So yeah. So that's what we talked about endoplasmic Golgi, plasma membrane endosomes. So you can see each one of them is recycling in a different in a different manner. Okay. All right. So now we'll talk about one thing. What are the emerging models of lipid transport? Okay. So this is very important slide. Okay. So so here what we want to talk about is so first of all, okay, so the first step here is that the phosphatidylcholine, the red color here, is synthesized on the cytosolic surface of the endoplasmic reticulum. Okay, and now it's freely flips across the ER membrane. Okay, now this gets diffused laterally in any membrane, and it goes the direction this way. Okay, and now so it gets diffused in any membrane, it gets diffused laterally. Okay, but now the phosphatidylcholine now travels to the what you call it travels through the vesicles. These are vesicles. It travels through the vesicles to the Golgi. So now it's traveling. So this red one here is the phosphatidylcholine. Okay, so they travel to the Golgi, and then from the Golgi to the plasma membrane. You can see how the red colors, how the red little blobs, they're just moving towards different organelle. So now it goes to the plasma membrane, and then now from plasma membrane to the endosomes. Okay. All right, so now what's the next step? So next step would be, okay, so then once it goes to endosomes, then it gets transferred by a transfer proteins between the cytosolic surface of the organelles. Okay, so that's now step number 5. Where's step number 5? Here's a 5. So that's a transfer is happening between the cytosolic surfaces of the organelles, and maybe sometimes through contact sites. Like for example, this is an ER. And you have the mitochondria through contact sites here. Okay, those are happening here. Now in the step 7, okay, so here's the step 7 where you have the ceramide. There's a ceramide transfer is happening via the transporter called CER or T transporters. Okay, from the ER to Golgi exchanges. Okay, and where there is a where the luminal sphingomyelin synthesis is happening. Okay, so. But however, keep in mind the sphingomyelin cannot flip, and what they do is they travel in the luminal leaflet of the vesicle. So basically, the sphingomyelin will not flip anything, but through vesicles, these ceramides, they travel via the luminal leaflet of the vesicle. Okay, so there are 7 different steps we talked about. Okay, the first step, again, let's summarize it again. The first step, you can see the phosphatidylcholine on the cytosolic surface of the ER, and that gets laterally diffused in the membrane. Then it also goes to the Golgi. It also goes to the plasma membrane, then to the endosome. From the endosome, it gets transported between the cytosolic surfaces of the organelles. So that's the number 5 here. And then through contact sites also exchanges can happen. Then you also have a ceramides that's where it's a transfer protein called CERT transporters that actually allows the lipids to take from the ER to the Golgi as well, basically for the luminal sphingomyelin synthesis. Okay, so but sphingomyelin cannot flip over, but they can also travel in the luminal leaflet of the vesicle. Okay, because it doesn't get flipped over, but it can still travel to the luminal leaflet of the vesicle. So now here, if you look at this, these are the principal pathways for the production of sphingolipids, sterols, and phospholipids. You can see here, these are sterols. These blue ones are sphingolipids, and then these ones are all in the mitochondria. But now these all are phospholipids. You can see here, it starts from the cytosol, that's acetyl-CoA. All the pathways gets inside. Okay, so here all the red color here, they all are ER. All the orange color here, they all are mitochondria. Okay, and then you also have the Golgi in the blue color. So you can see now how this whole machinery of the pathways are going from one organelle to another organelle. Okay, so cytosol to ER to Golgi to mitochondria. So much of exchanges are happening. Okay, and you can see it starts from acetyl-CoA, and in the mitochondria it ends up with the phosphorethanolamine and phosphorylserine. okay? Whereas in the case of Golgi, you can see still there's some phosphatidylcholine and phosphoethanolamine is being synthesized, but it also will be useful for the sphingomyelin synthesis here. Okay, so basically this is showing how the exchanges are happening between lipids biosynthesis. Okay, it starts from cytosol, but it's getting synthesized in various manner in different compartments. So this is how the lipid exchanges can occur very vigorously in the cell. Okay, you guys follow? So what is the confusion? So what do you guys want to know more clarification? So is there any confusion here? We can talk about it if something is unclear. You guys clear here so far? Guys?
Okay, are we learning about how the membrane is synthesized or how the lipid is synthesized?
So how the lipids are being exchanging. Okay, so it's not about synthesis. You can see it starts from acetyl-CoA, right? I mean, there's a lot of things that are there, okay? These are very sequential process, right? So each one of them, if you look at it very carefully, for example, cholesterol. Okay, but the cholesterol doesn't get synthesized here just by itself in ER. Does it? No. It starts from where? So if you go backtrack them, it all starts from acetyl-CoA from cytosol. Yeah. So there is an exchange between two organelles, cytosol to ER. Yeah.
Yes.
Okay. So same way, if I ask the question, phosphatidylserine, does phosphatidylserine is being produced just by itself in mitochondria?
No.
So where is it coming from?
The pink one.
Yeah, which is the ER.
ER.
Yeah, but if you backtrack them, this phosphatidylserine is coming from diacylglycerol. Yeah.
Yeah.
And diacylglycerol to phosphoroacetic acid. Okay, and then it goes back here. Okay, and then it gets inside here. Some of this phospholipids also converts ATP, and the PIP3 is the triphosphatyl inositol phosphate. It's being synthesized in the Golgi. I mean, we don't have to go into all those details, but the general take-home message here is that the phospholipids are not being synthesized just by itself in one specific organelle. It's actually an exchange between two different organelles, because it's a sequential process of different pathways involved. So that's why I keep telling there's always an exchange is happening between two different organelles. So for example, so here's one example where the CL is another lipid. Okay, so now there's also let's talk about phosphoethanolamine here. Okay, that's another lipid here. But that doesn't get synthesized just like that, am I right? Because it's converting from serine to ethanolamine. But that ethanolamine, once it's being synthesized, it's also getting inside the ER. All right. Because you see the arrow, just follow the arrows. Okay. So the arrow shows where the translocation is happening, where the transport is happening. Yeah. So the point here is that, all the synthesis, like on another example, if I want to tell you, cholesterol. Okay, so there's also cholesterol being formed here too. There's also cholesterol being formed here too. So there are various ways in how a product can be formed. You know what I mean? Okay, so here you can see, if you look at this here, the arrows goes from this bidirectional. So basically acetyl-CoA to to triacylglycerol is going. There's also end product called cholesterol. Yeah. So the whole point here is that two important points. Okay, the phospholipids are not being synthesized just by itself in one specific organelle, because the phospholipids are being synthesized while doing the exchanges happening between two different organelles, like which means, it starts from cytosol and something is being produced in ER, and that from ER it takes to the mitochondria, or it takes to the Golgi, and so on and so forth. You know what I mean? So
I have a question.
Yes, go ahead. Yeah.
So the phospholipids, that's part of membranes. So are we saying that acetylcholine is in a membrane, and that membrane interacts with ER, which interacts with?
Lipids, they are embedded in the membrane, but it doesn't mean acetyl-CoA has to be synthesized in the membrane. Acetyl-CoA is being synthesized in the cytosol.
Okay.
Okay, but the derivatives of acetyl-CoA is being sucked up by the other organelle for its own synthesis. You know what I mean?
Okay.
Yeah. So that's why, I showed you in the previous slides, where we talked about exchanges are happening between phospholipids. If you look at these slides, if you look at this slide, this is what I was showing. You see, this is cytosol, yeah. So this is outside, yeah. And what I'm showing is the ER here, yeah. So this is a bilayer. I mean, not bilayer. This is bilayer leaflets, but it's bidirectional. Okay, so which means, the piece, these are symmetrical, am I right? Because PC to PC, P to P, which means, like, whatever is being synthesized here, it goes to cytosol. You understand? But that's not the case always in the Golgi, because if you look at this one, only PE and PS are being translocated to the cytosol because of the P4 ATPases. But whereas the rest of them, most of them are present within the luminal leaflet of the Golgi itself. They're not going outside. So this all boils down to how the lipids are being translocating between different organelles, and what is being consumed and what is being synthesized. Okay, so in order for mitochondria to generate the phosphorylserine or phosphoethanolamine, it has to, so phosphorylserine or phosphoethanolamine, it has to depend on other processes here. Do you follow?
Yes.
Yeah. So that's what I was trying to say. So there's a constant exchange between phospholipid. This is why I keep talking to you guys about lipid exchanges. Okay, and synthesis of the lipids also can occur, and which we talked about it, because, as you know, if you look at this structure here, if you look at this one, where do you see majority of the lipids are being synthesized? In which organelle?
Golgi.
Why would you say that?
Okay, that's just where I see the last result.
Okay, where do you see more lipids are being present?
ER.
That's right. So that's why they say ER is the main site of phospholipid synthesis, because you can see majority of the lipid reactions here, what they're showing, they all are in the ER.
This is going to be a dumb question, but is acetyl-CoA a lipid?
Which one? The phosphate. Where is the acetylcholine is there?
Just acetyl-CoA. Because in my head, that's where I'm blanking, because I don't think acetyl-CoA is a lipid.
Here, the acetyl-CoA is being taken in from the cytosol. You see here?
Yes.
But it's converting them into HMG-CoA, okay?
Yes.
So and then it goes to mevalonate, and mevalonate to all the cholesterol pathway.
Okay. Cholesterol is a lipid.
Yes.
Okay.
Okay. So the point here is that, if we just take a step back and look at all the slides that we talked about so far. I talked about the slides where I showed you where the main site of phospholipids are. ER, we talked about. We also talked about how the lipid exchanges are happening between different compartments, like Golgi, endosomes, plasma membrane, and ER. We talked about it. Yeah, but not all the phospholipids are being translocated to the cytosol. The one is some, but not all. We talked about it. Here's one slide to summarize further that how the actual biosynthetic pathways are happening. Okay. So and kind of like it all ties up very nicely, to understand. I know there's a lot of information here. I'm not asking you guys to memorize all this information, but I think the general take-home message is how the membrane lipid biosynthesis are being exchanging, between different organelles, and how the sequential events are there, So that's what I want you guys to follow up here. Okay. So a couple more slides, we'll talk about it. This is very important. Okay, so the lipid composition sensors. Okay, so basically there's a gene called IRE1. IRE1 stands for a membrane fluidity controlled by ER stress sensor called IRE1. So the IRE1 is a conserved endoplasmic reticulum resident stress sensor. So that can trigger an unfolded protein response. So UPR. UPR stands for unfolded protein response. Okay. So through a luminal domain that monitors the unfolded protein that is loaded into the endoplasmic reticulum. Okay. So basically the IRE1 uses its membrane span to detect membrane perturbations. Okay. Such as, for example, if there's an increase in the lipid saturation. Okay. So this can signal the unfolded protein response gene independently of the luminal stress sensing domain. Okay, so you can see there's a normal signal, it's a saturated lipid stress, there's a restoration to normal. So we'll talk about it, how the IRE1 can actually sense, under stressful condition, how does this trigger the UPR response and bring the membrane fluidity to the normal state. Okay, so here's one. First, on activation, IRE1 is autophosphorylated and assembles into a higher order oligomers. Okay, so here's what autophosphorylation happens there. Okay, the P stands for phosphorylation. Okay, it's autophosphorylation. Now, the signaling usually proceeds through a splicing event called the XB1 mRNA. Okay, so this is initiated by the IRE1 cytoplasmic RNase domain. Okay. So only the spliced mRNA is translated to produce a transcriptional activator called the UPR, the unfolded protein response genes, okay, which code for the folding enzymes, okay. And now what happens? Now it allows, okay, the ER membrane to restore its own homeostasis, okay. So this is how the normal situation happens. But under stressful condition, the IRE1 autophosphorylates it, but this is usually being initiated by the IRE1 cytoplasmic RNase domain. Now the spliced mRNA is translated to activate the transcription of this UPR genes, the unfolded protein response, to restore back the stress signal to a normal. Do you guys follow? So which means you have a normal, you have a lipid stress. Under lipid stress, what happens? Because under lipid stress, there's a lot of disturbed membrane perturbations will happen. But the IRE1's job is to protect the membrane perturbation to happen so that the normal restoration of the lipids can happen, okay? Because you need to bring back the normal ER homeostasis. Is that right? So under stressful condition, what happens? There is an autophosphorylation will happen. The autophosphorylation will lead to a transcription of this activator called UPR genes, but that can happen only when you have a spliced mRNA that gets that needs to be translated. Okay, and that allows for a proper folding of the enzymes, which leads to a proper restoration. Do you guys follow? Do you guys follow or not? You guys are keeping quiet.
So
I don't know. Go ahead, Alok. Sorry.
No,
Yeah.
you can go.
Go ahead.
I was just going to say in general, I'm a little lost, but
okay.
Alok, go ahead.
All right. Okay. Let's take a step back. Okay. So under lipid stress condition, what happens under lipid stress condition? So under lipid stress condition, what happens?
The membrane becomes saturated.
Membrane becomes saturated or membrane becomes perturbed? Because under stressful lipid stress, a lipid stress. So under stressful condition, what happens to the lipids is what the question is. So under lipid stressful condition, there's an increase in the lipid saturation. That means there's an aggregation that allows to create a membrane perturbation or membrane impairment. Okay, because it's too much, it's okay. All right, so let me take a step back. Okay, so okay, so how do I put it? Okay, if you keep eating the sugar for 10 days, okay, like 2 kilograms of sugar, okay, is it good for you? Why? Because your body cannot metabolize it. Yeah, it's too much. Yeah, same thing. Anything is too much is toxic to the cell. Yeah, so which means if you have an increase in the lipid saturation because it's too much of lipid, then the membrane will almost, it will sense it as though there's a perturbation, there's an impairment of the membrane is happening, because it will break down. The membranes will perturb in some fashion, you know. Okay, so because membrane structure, you know, has to be intact. Am I right? Membrane cannot be broken down. So IRE1's role is to it'll span the membranes, because IRE1 role is actually ER membrane, because it spans the membrane, and it actually makes sure that, you know, the perturbations is not happening, because membrane architecture has to be maintained in order for the normal homeostasis to happen. You understand? If a membrane gets broken, how can you have a normal homeostasis? You cannot have a proper homeostasis. What does a homeostasis mean? A balance, am I right? A balance in the membrane structure. So you cannot have a membrane perturbation. If you have a membrane perturbation, then the ER pretty much, you know, the homeostasis is lost. If the ER homeostasis is lost, then pretty much, you know, the cell gets messed up. Okay, so what the point here is that how is the membrane fluidity is being controlled? So why membrane fluidity is very important? Because the membrane fluidity is very important because that's where the exchange is happening. A lot of exchanges are happening between organelles. Am I right? So the IRE1's role is a sensor. It detects if there's a perturbation. The perturbation can happen because there's an oversaturation of lipids. Okay, or the perturbations can happen because when there's a stress in the lipids. Do you understand? Okay, so, and that's what, so in order for the membrane fluidity to be controlled, and so that there's a proper ER homeostasis, the IRE1's job is to sense where the perturbations are happening. It's kind of like a foreign, it's kind of like a protector, okay, a protector of, you know, of any disturbances. Okay, so it wants to make sure the homeostasis is maintained. So what they are trying to say is that if there is a lipid stress is happening, okay, that leads to excess lipid saturation, then the first thing the IRE1 does is actually it autophosphorylates. It precisely autophosphorylates on what they are saying here on the IRE1 cytoplasmic RNase domain. That's where it starts initiating. You see the little p there? And that's autophosphorylation, okay? But this autophosphorylation can trigger, okay, the spliced mRNA, okay? Because you see, why does it splice? Because the XB1 is a major player of the unsaturated lipid synthesis, because basically it doesn't allow, so the XB1 has to be maintained properly, because anything with a more saturation leads to the XB1, because the XB1 plays a major role with the saturation here, and that might lead to the perturbations. So the first job is for IRE1 is to splice that XB1, okay, and then because it tends to activate the important gene called unfolded protein response, because you need to make the protein to be folded properly in order for the homeostasis to happen properly. So, and the job here for the IRE1 is to activate this, what is called unfolded protein response genes. Okay, and this unfolded protein response genes will lead to the restoration of the ER homeostasis back. Do you guys follow?
I think my confusion is I thought you described what it's going, which protein it's going to unfold, but you're just saying it'll lead to the restored normal.
Yes, because you need to bring back the to the normal state. Am I right? So restoration means if there is a disturbance in the membrane dynamics, you need to bring back the disturbed membrane to its normal state. That's what it means.
By unfolding proteins.
That's right, because the unfolded protein response genes has to be activated. Because the unfolded protein response genes are the ones that controls majority of the homeostasis.
Oh, okay.
Okay, so this is the only way where the IRE1 can restore back the ER homeostasis or the membrane homeostasis back. So it has to activate the unfolded. Don't get confused about the word called unfolded protein response genes. That's how it's called. Okay, so the UPR genes can be under stressful conditions. These genes needs to be activated. Okay, otherwise there's no way, because this UPR genes, if they're not activated, then there won't be any proper folding structure happening. Okay, because the main purpose of the UPR genes is to, many of the UPR genes, mostly they are actually called mitochondrial UPR genes. Okay, but anyway, don't go over there. But the UPR genes' role is to bring back the disturbed membrane to its normal state. Okay. I think Thomas might have some questions, or what, Thomas?
No, I think I'm following better now. The role of UPR is mostly what I was confused with, but that makes more sense.
Yeah. Okay. So, but keep in mind, okay, the autophosphorylations can happen. It doesn't. The autophosphorylations can always happen. Under stressful, not always, but there are some genes that activates this autophosphorylation as a process of bringing back some machineries back to its normal state. Because without autophosphorylation, because IRE1 is a sensor, the first defense mechanism is to activate the phosphorylation, okay, through autophosphorylation. Okay, do you guys follow? Okay, and then it goes through the XB1s and all those things, and then activate the UPR, and then restore back restore the lipid stress molecules to its normal homeostasis is what is happening here. Okay. Okay. Yeah.
The one thing I'm wondering is there not a feedback inhibition before we get to this point to slow down the production of these lipids that are oversaturating the membrane?
Well, that's true, but unless there are some other machineries out there, which I am not sure, but it's quite possible. But usually, these events could be a dynamic event, or I don't think the lipids could be a very slow event, because there's a lot of organelles are dependent on this lipids, so it should be a very dynamic process. You know what I mean? So the aggregation, there's no way. I mean, unless there might be something else would be there, but the aggregation event, cannot be a slow process. It has to be a dynamic process. Given the fact that how these lipids are very important and used by other organelles. You know what I mean? So it cannot be a slow process.
Yeah, so you're getting like a rapid influx of these lipids from
Yeah.
aggregation with some other organelle or some other
Organelle,
membrane, something
exactly,
like that, and
exactly.
giving you this rapid
I
response.
mean, it's also important, stress, the stress can be, the physiological cues can be n number of factors, there's not one factor, the lipid saturated stress is always based on n number of factors, it could be disturbance in the pH level, or it can be disturbance in, whatnot, mutation, I don't know, whatnot. I mean, anything can lead to any level of stress, but here we're talking about a function of a sensor molecule called IRE1. What does the job is? You know what I mean? Okay, so to summarize it, so we just went through a lot of things here in this class. Okay, so we went with the different lipids, we talked about triglycerols, we talked about functions of so many other lipids, and we talked about where the lipids are being synthesized and how they are translocated to different organelle. And we looked at the crosstalk between organelle pathway, like how Golgi, ER, and Golgi, cytosol, how the lipids are being exchanged, are being synthesized. And we talked about IRE1 sensor as an example to see how the lipid compositions or lipid stress molecules are being restored back to its normal state. So in general, we covered all those things. So, if you guys have any questions, I think Brian Phillips, I think she raised her, he raised a hand. I don't know. Anything that, what is in the chat box? Yeah, the unspliced form of XB1 acts as a feedback inhibitor. So unless IRE1 splice, it inhibits URP. Okay, that's fair enough. That's possible. Yes. So what's the question here? So it's URP responding to unfolded proteins in the membrane, or is it directly? It's URP is responding to the unfolded proteins. That's what it's responding to. Okay. XBR1 is a transcription that activates UPR. That's fine. Okay, what is this one new message? So I don't understand. What does it mean? Causes to fix lipids. So what? Yes, it fixed the composition of the membrane. Yes, so that's why it can restore the membrane the membrane back to its normal state without causing any perturbations. Yes, that's what it is. Okay, so we will leave it at that, and I think we will continue the next lecture. So, but if you guys have some questions, we can talk more about it next class. Okay, but you guys are following the lecture so far. I mean, this is like so why does it respond to unfolded protein and goes ahead to change the lipid composition? Well, that's a good question. So maybe there might be other genes that might be involved, in the sequential process, which we may not know. We may not know about it. I think that's quite possible. So I don't think the lipid composition is being changed. Okay, I think it's not the composition of the lipid is being changed. I think it's just a restoration of the membrane. I think that's what it is. Okay, the restoration of the membrane means the lipid balance is there, which means there's because the UPR is being activating as a transcriptional factor. So it activates the genes. So maybe there's more transcript levels out there that balances out the composition of the lipids, I suppose. So we can discuss more about this, in the next class. I think but these are very good questions. So yeah. Okay, so when are we getting the questions for test? Well, when is the exam, by the way? The first midterm.
I think first midterm is October 6th. That's Wednesday of not next week, but the week after.
Okay, so next week we can talk about that, and we can take some questions and we can go from there. Yeah. So next week, half a class, we can review the other lecture, and then we can discuss the questions, and then we will come up with the list of questions, and then we can go from there. So I think that should be doable next week. Okay, so so yeah, so I think these are good questions. So we can talk more about it next class. Okay, so and I think the next class we will talk about proteins, and then we'll spend half a class after 7:30. We can spend looking over the questions and see how we can set up the midterm questions. Okay, all right. Okay, all right, guys. You take care. So we will. Next class, I'll get another webcam, like third one. They break so easy. Okay, all right, fine. Okay, all right. Okay, so we'll keep in touch. So next week we will meet and we'll go over the lectures and the questions as well, guys, for the midterm. Okay, all right. You guys take care. We'll talk soon.